And so just briefly about some of the topics I'm going to be covering today. I'm going to first start off with our kind of flagship project called SCADE, or Single Cell Atlas of Immunological Diseases. And then I'm going to go a bit into a few GeoMx projects that we've done in our lab, and then kind of end with a kind of bigger picture GeoMx plus single cell RNA sequencing multimodal analysis across diseases. And so, right, to first start off, so SCADE, or Single Cell Atlas of Immunological Diseases, is a program funded by the Korean government where the goal of multimodal high-dimensional profiling of 16 plus target immunological diseases with unmet medical needs. And these diseases can range from rheumatoid arthritis, from inflammatory bowel disease, from CNS inflammatory demyelinating disease such as multiple sclerosis, to atopic dermatitis, psoriasis, and so on. And so the goal is to profile these diseases and collect all of these single cell data sets, including over 37 million cells collected. And this is part of over 24 research groups that are in collaboration with this project. And so a brief layout as to this project, as some of you may have heard already, the first layer of this project is to generate a single cell transcriptome atlas, where we collect single cell samples across all of these different diseases that I just mentioned, and we profile these diseases. And then we incorporate a second layer with a proteome atlas, including single cell proteome data, including mass cytometry or CyTOF. And then a third layer with single cell epigenome atlas using ATAC sequencing to get into the epigenome information. And then finally, more related to this talk today, is the fourth layer where we'll incorporate spatial transcriptomic and proteomic profiling of these target diseases. And in particular, we plan to use the nanoString GeoMx platform for spatial proteomics, and then also the nanoString GeoMx platform for the spatial transcriptomics. And so being able to utilize both modalities is very essential into helping build this immunological disease atlas, and then to hopefully identify and validate potential therapeutic molecular targets of these inflammatory diseases. And so the way that the project is structured is there are clinical cores that we have collaboration with all across the nation. These physician scientists will collect patient samples. They'll collect either tissue organ samples that will be sent to the tissue processing cores, or they'll be sending PBMC blood samples, which will be sent to PBMC processing core. And then each of their respective PIs and physicians will conduct their own disease-specific analyses. These will then all be transferred into the meta-analysis core here at the Genomic Medicine Institute, SMU, also with clinical information from the clinical core, and that will all be aggregated here, while we're performing meta-analysis across all of these different diseases. And so the meta-analysis core is being led by Dr. Jong Kim, from the director of the Genomic Medicine Institute, the PBMC core by Dr. Jong Park and Dr. Hyunjae Kim, and then the tissue core by Dr. Dong-gi Kim and Dr. Jung Seok Lee. And so with that project, because of real importance is that spatial aspect, with GeoMx, we wanted to be able to do first kind of single disease targeting of using GeoMx. And so I'm first going to talk about a few different GeoMx platforms, experiments and projects that we've done currently or that are ongoing. And so just a few of these is triple negative breast cancer, hidradenitis suppurativa, and hepatocellular carcinoma. And so our group right now using the GeoMx platform has over 28 main projects generating GeoMx data, where we've collected over 2,000 ROIs from the GeoMx platform. And we also have included matched single-cell RNA-seq data sets from the same patients, and additionally from other projects for further validation and for reference. And so obviously I'm not going to have time to speak all about all these today, so I'll just speak about a few of these projects. And so I don't think I need to really go into the details, as I'm sure many of you already know very well what the GeoMx workflow entails. But just to briefly recap how it works is we collect sample biopsies, either in FFP or frozen. We then section those onto slides, and then those slides are subjected to UV photocleavable oligos and fluorescent antibodies. That slide will then go into the GeoMx instrument, where the fluorescent antibodies will help to determine which regions of interest will be selected for GeoMx profiling. Those oligos will then be cleaved will be UV cleaved and then dispensed per ROI into a 96-well plate, where that plate is then sent off for sequencing to identify all of the transcriptomic information from each single ROI. And so the first project that I'll talk about is a triple-negative breast cancer project that we're currently working on. This is where we've collected tumor and draining lymph node biopsies from both responders and non-responder TNBC patients who were enrolled onto neoadjuvant immune checkpoint blockade plus cytotoxic chemotherapy. And so what we wanted to do is to really compare gene signatures and pathways associated with responders and non-responders from both the tumor and importantly, where all the immune priming is going on in the draining lymph nodes. And then we also wanted to compare CD4 T cell, CD8 T cell, and CD8 macrophage cell type signatures and gene signatures between these responders and non-responders to get a better sense of why patients are responding to neoadjuvant treatment and why they are not. And so as a pilot study, we've collected 4 breast cancer tissues, each with 12 ROIs, and then 3 lymph node slides as well, each with 12 ROIs. And so after collecting these ROIs from the respective images, we subjected these to differential expressed gene analysis. And so we wanted to compare the gene sets between responders and non-responders. And so while I won't be able to show exactly which genes they are today, we've generated a gene signature that can help predict whether these patients will respond or not respond to ICB treatment. And not only that, we used a ROI cell type deconvolution algorithm because these are ROIs and not single cell targets. There's going to be dozens or hundreds of cells within a single ROI. And so we wanted to be able to predict, based off the gene signatures within that ROI, what are the different proportions or frequencies of cell types within a single ROI. And so we use a deconvolution algorithm to essentially predict the different types of immune cells that are within a single ROI. And what's quite interesting was that when we compared the frequencies of these predicted cell subsets, we found a significant increase in naive B cells in responders. And then we also found a significant increase in macrophages and CD4 Treg populations within non-responders. And so particularly the CD4 Treg phenotype is quite well known across various cancers where the CD4 Treg will play an immunosuppressive role and how a pro-tumorigenic role. And so this was a nice kind of validation of that. And then we also want to phenotype a bit more these macrophage populations to see whether they're playing an inflammatory role or a non-inflammatory role, or maybe a more M1 or M2 screwed role. And so based off these initial findings from this initial ROI GeoMx data set, we wanted to further validate this phenotype. And so now we're planning to use not just the transcriptome information from GeoMx, but we also want to take subsequent slide sections and then subject them to the GeoMx protein assay, where we'll then hopefully be able to better characterize subsets of macrophages and subsets of CD4 Tregs and B cells that are correlated with response or non-response in these TNBC patients. And so with that, just quickly, I'd like to thank Professor Sung-Baek Kim and Dr. Jae-won Hyung, who are the main leaders of this project, collecting patients and then sending them over to us for GeoMx analysis. Next, I'd like to skip over to a completely different disease area, hidradenitis suppurativa, or HS. And so HS is a chronic inflammatory skin disorder that causes these deep and painful lumps or abscesses within the skin. And so they're characterized by these kind of deep nodules within the skin. And so the goal of this project was to compare HS with epidermal cysts. So in HS, these cysts are characterized by these inflammatory sites, whereas epidermal cysts do not have these inflammatory sites. And so we wanted to kind of understand the disease pathogenesis of early stage HS, comparing the inflammatory sites within these cysts versus non-inflammatory cysts. And so we collected ROIs from both the epithelial regions within HS and also within kind of immune-infiltrated areas of HS compared to these epidermal cysts. And so one of the main findings when we compared epidermal cysts versus early HS was when we subjected these ROIs to gene pathway analysis, we found, first of all, that neutrophil degranulation was heavily enriched in HS. Also, importantly, signaling by interleukins was also enriched, and also response to bacterium was heavily enriched in HS. And so we wanted to interrogate this phenotype a little bit further. And so some of the genes related to commensal bacteria we saw were highly elevated in HS, suggesting that this might play an essential role in the early pathogenesis of this disease. We also looked within the epithelial cells and saw an increase in IL-13 receptor alpha-1 and IL-4 receptor. And so these receptors are all kind of receptors for Th2 related cytokines, suggesting that there's an increase in Th2 cytokine sensitivity within epithelial cells, suggesting that this may play an important role in the pathogenesis of MS or of HS. And then also another important cytokine, IL-36G, was also elevated. And so IL-36G is known to cause neutrophil degranulation, generating these kind of net neutrophil extracellular traps. And so this netosis can then amplify the inflammatory response within the epidermal nodules, causing this kind of positive feedback loop of increased inflammation, leading to disease progression. So we think that plays a very important role in early onset of HS and then later on as the disease progresses. Similarly, with it, we also see that it's not just IL-36G, but also downstream where we actually see neutrophil degranulation related pathway genes that are also all heavily increased within HS by using the GeoMx platform. And so finally, also, we wanted to look at the immune cell compartments collected by GeoMx. And when we compared HS to epidermal cysts, we found these 126 genes that are upregulated within HS. And then in particular, many of these genes were kind of B-cell related genes, where we saw an elevation in many of these B-cell related genes in these durable infiltrating immune cells, suggesting that B cells are also having an important role in the pathogenesis of HS. And so this paper was just recently published in the British Journal of Dermatology in collaboration with Pil Jin and Jung Shin and James G. Kruger, where we've just reported kind of these findings very recently. And so I'd like to thank Pil Jin, Jung Shin, both in the Department of Dermatology, Pil Jin from Seoul National University College of Medicine, and then Dr. Jung Shin from Chabundam Medical Center, as well as Christine and Soyoung from our lab who really led this project and got it to be published. And then finally, since I'm not going to have too much time to talk about this last project, I just wanted to briefly mention a project where we had both paired single-cell 10x data, also with paired GeoMx data from the same patients. And so this was a hepatocellular carcinoma project where we were going to profile HCC liver biopsies from patients who had an early relapse in HCC compared to just primary HCC, and to identify the key crosstalk interactions that are happening between the tumor cells and the actual tumor environment within HCC. And so one of the findings that we identified by single-cell RNA sequencing was that these mate cells, or these mucosal-associated invariant T cells, were significantly increased within patients with early relapse HCC compared to primary HCC. And so now we're using GeoMx to kind of identify that phenotype, to validate that phenotype within the actual section itself, and then to identify immune cells or other non-immune cells that are closely interacting or within the vicinity of these mate T cells to identify the exact kind of cell-to-cell communication that is occurring and the reason for why these mate cells are playing a role in early relapse of HCC. And so this project was led by Dr. Jaelan Lee, who's in the Department of Radiology here at Seoul National University Bundang Hospital, and then led by Tui, Hayoung, and then Konu from our lab. Okay, and then going on to the actual, so each of these projects so far has been just kind of single disease-related topics. But now that we have so many different projects and so many different diseases, I was going to go now a bit more into actual multimodal analysis, not just from one disease, but across diseases using the GeoMx platform and the single-cell RNA-seq, the split-seq platform. And so just a brief overview of the study design. So this is by split-seq. We collected 20 IBD patients, 20 AD patients, 20 psoriasis patients, and 20 COVID vaccination patients, as well as 14 matched healthy donors. We then subjected each of these PBMC samples to a 4-hour PMA plus ionomycin stimulation to stimulate T cells. We then performed split-seq. And so split-seq is a single-cell RNA sequencing method, very similar, well, it's a single-cell RNA sequencing method that, unlike 10x, where 10x uses a gem emulsion bead to capture single cells, split-seq actually uses combinatorial barcoding using a well-based barcoding system, where essentially 4 barcodes are ligated to a single cell, allowing for single cell characterization of samples. And so the idea is you take all of these 96 samples and you first barcode, you reverse transcribe with the first barcode. So each sample is labeled into a 96-well plate. That sample is then subjected to a first barcode. And then these are all pooled together and then split again, and then ligated with a second barcode. Then all the samples are pooled again and split and ligated with a third barcode, pooled again and split and bound with a fourth barcode. And so essentially, by having these 4 different combinatorial barcodes, the chance of a single cell having the same barcode becomes very low, and it's why we're able to identify 1 million cells within this project. And so the goal of this project was to identify for the identification and the functional characterization of disease-specific pathogenic immune cell types from activated PBMCs that are otherwise difficult to delineate at resting conditions. And so many times when we work with PBMC samples, they've been cryopreserved and then maybe they've been rested, but most times they're at resting state. And so many times these pathogenic signatures that may be occurring in these diseases may not be essentially well identified within the resting state. But when we subject them to activation by PMA ionomycin, it kind of we're able to better identify these pathogenic cell types. And so once we've got sequencing data back, this is all 1 million cells. And I first just kind of annotated broad cell types like B cells, dendritic cells, monocytes, NK cells, NKT cells, T cells, and PDCs. And then this is the UMAP of kind of the different diseases. And so after the annotation of these, I really wanted to focus on the NKT cell and T cell populations. And so I took all those T cells and then did subclustering of those T cells. And what you can see, interestingly, is that each disease compartment has a specific enrichment of different cell types. And so this is just a kernel density UMAP, where we can see that COVID has an enrichment in these specific T cell subsets. Atopic dermatitis has a enrichment in these specific disease subsets, T cell subsets. IBD has enrichment here, and then psoriasis has an enrichment of T cell subsets within here. And so we wanted to go a little bit further to characterize these. And so we first did subclustering of those T cells, and then we performed DEG analysis of those subclusters. And what we found particularly was that subclusters 5, 9, and 15 all had high expression of this particular gene X. And what you'll notice is that 5, 9, and 15 are all kind of grouped together here within this UMAP. And if you remember, T cell cluster 5, 9, and 15 are all enriched within psoriasis. And so we also wanted to look at the frequency of these clusters. And so indeed enough, and we take the same number of T cells from each disease group, from AD, COVID, psoriasis, IBD, and healthy controls. What we see is that clusters 5, 9, and 15 are all completely enriched by psoriasis samples. And so the frequency of these subclusters are all enriched in psoriasis. And so when we look at the gene expression of that particular gene X, we can also see that it's highly enriched within the psoriasis-specific T cell cluster. And so we think we've identified a very psoriasis-specific activated gene signature that may help delineate and identify psoriasis, and then also identify these pathogenic T cells that are very specific to psoriasis. And what was even more interesting now is going into the GeoMx data is we had a project where we collected various different diseases, mostly including skin diseases, including psoriasis and atopic dermatitis. But we also had some patient samples that had an overlap in phenotype. And so there were some patients who had a very exact AD phenotype, some patients who had a very psoriasis phenotype, and then there are also some patients who had a kind of AD and psoriasis combined phenotype. And so we wanted to understand, is this just at the phenotypic level, like just at the skin, just looking at the skin, or is this actually, can we see that recapitulated within the actual transcriptomic and gene levels? Is there overlap between gene expression data? Are there common features between these diseases? And so what we did is we took skin biopsies from atopic dermatitis patients. We took skin biopsies from these kind of AD and psoriasis overlapped skin patients, and also skin biopsies from these psoriasis patients. And then we performed GeoMx on each of these skin biopsy sections. And what was very interesting was that, so here in green is the AD ROIs, and this is looking at CD45, so immune infiltrated ROIs. And what we'll see is that that same gene X that I mentioned that we found identified in very specific to psoriasis, that gene is lowest in AD, and that gene is also highest in psoriasis by GeoMx. And then also there's a very interesting kind of correlation of this gene. And so this mixed type 1 immune cell in red is a section that was collected again from these patients who had kind of both AD and psoriasis-like features. But this was a bit more closely related to AD as determined by the physician. And then this mixed type 2 subset was a bit more closely related to psoriasis, but also had some AD features as again determined by a physician. And so we can see is that correlates exactly with the expression of gene X. So as its expression of gene X increases, the phenotype towards psoriasis at the transcriptomic level also increases. And so we think that this gene X is very important, either in identifying or in its role in the pathogenesis of psoriasis. That's also recapitulated with this kind of AD psoriasis overlap in psoriasis data set that we found by GeoMx. And so we were essentially able to validate this gene, not just with one platform, not just with SplitSeq, but we are also able to validate that platform, not just in circulation, but also within the skin, local microenvironment. And we also conducted a separate 10x single cell experiment on skin biopsies, where we also find that this same exact gene X is also upregulated in psoriasis. And so we have kind of 3 independent different modal platforms that have all independently identified the upregulation of gene X in psoriasis. And so now our goal is to further characterize what exactly this gene, what the role of this gene is in the pathogenesis of psoriasis, and then also going into more of the spatial features. So what cell types that express gene X, what are they closely linked to? Are they cross-talking? Are they interacting with a certain cell type that's neighboring these genes? This is all planned to be doing with additional GeoMx profiling and also other spatial profiling platforms. And so with that, I'd like to thank Dr. Jung Kim from Hanyang University, the Department of Dermatology, and also Dr. Simfil Jin from Seoul National University College of Medicine, the Department of Dermatology. And then also Hyojung Nam, Kim Cho, and Hyun-Sung Che. They're all students in our lab who helped, who all made the GeoMx data for this project. And so that was just kind of one example of how we can a few diseases and then try to find interactions between those diseases, so do multimodal analysis and multi-disease analysis. But now our goal is to kind of expand that analysis. And so because we have so many ROIs, we have over 2,000 ROIs by GeoMx and over 20 diseased and healthy tissues, ranging from various cancers, skin, lung, kidney, heart, and brain immune-related diseases, to kind of integrate all of these ROIs together. And then also with single-cell RNA sequencing, with ATAC sequencing and proteomics, and also other spatial transcriptomic and proteomic platforms to kind of integrate all of these together and to be able to really map out disease, inflammatory diseases, and also cell types. And so one possible example is to perhaps characterize and understand the mechanisms of a specific cell type, maybe like Tregs, and their role across a wide variety of diseases and disease settings. And so the goal is kind of take all this data together, integrate all this data, use machine learning to create a model training method to then be able to identify these the role of these certain cell types across diseases, across tissues, and then importantly to be able to map the key cell-to-cell interactions that are happening with these cell types and their role in disease progression and neural development, and then to proceed on with actual endotyping of disease subcategories to get a better understanding of not just single disease progression, but the role of various cell types across various disease pathogenesis. And so we're also collaborating with Sarah Techman, with Aviv Regev, Miriam Murad from Mount Sinai, and James Cooper as well in this overall. And so with that, I'd like to give out a special thanks to our team, particularly my supervisors, Dr. Chung-Gi Park and Dr. Hyun-Jae Kim, and then also our entire lab, all of our students, our postdocs, and MDs and physicians who have all helped and have made this all project possible, and if I didn't have the help of this amazing group of students and physicians and scientists, none of this would have been possible. So thank you to everyone. And yeah, thank you for taking the time to listen to my talk today. And I'm open to take any questions.